Hello, welcome to the pre-party for Wednesday, September 25th. We're still in our soil, grains, and baked goods sections, although we are going to switch a little bit of a gear right now and going forward into Wednesdays, Fridays, and next week's topics. Instead of talking about carbohydrates like we have in the last week or so, we are going to switch to proteins. We are still in that baked goods section, so you may be wondering, well, do proteins really play a large role in baked goods? And in fact, they do. So that's why I'm going to bring it up now. We're going to continue talking about it through the semester. After today's pre-party, you should be prepared for the actual party and class we will have by knowing what flour and baked goods contain in terms of protein content. So we're going to look at a food label, understand that protein is identified on that food label, and there's a reason because that wheat kernel that has been our model for the last week actually does have some protein in its structures. Once we go beyond this, we are going to answer the question, what are proteins? Some of you may have a really good idea. Some of you may be new to the protein concept and really want to understand what they are beyond just maybe what you know, they're meat, egg, poultry, but they go beyond that. And then finally, we're going to get really interested in protein structure because on Wednesday at the party, you will be able to put together your creativity in forming protein structure. So here are two food labels, look familiar from other pre-parties that we've done, white flour and whole wheat flour. You'll notice that I've highlighted the protein component, white flour having 3 grams of protein, whole wheat flour having 4 grams of protein. So we know protein exists in flour, but where does it come from? Knowing that flour is primarily endosperm because the endosperm makes up 83% of the wheat kernel, we understand that it has starch in it. We know that from last week's activities. But if we look at the endosperm more closely, there are actually is a large share of protein found in that endosperm. So that's why we see it in whole wheat and white flour. If we're looking at whole wheat in particular, when we include the bran and the germ, there are still protein components in each of those. So that's probably why we have 4 grams of protein on the whole wheat flour label versus 3 on the white flour. So we know a wheat kernel contributes protein to flour. Well, what does that protein actually do when we consume it in the form of a baked good or anything that we're including from a carbohydrate source, such as flour? In the body, protein has many functions. There are far too many for me to list. I've just listed a few here. A couple that I want to point out, though. Our body runs on hormone regulation and the activity of enzymes. Enzymes being something that can make something happen in our body. Those hormones and those enzymes are all made of protein. So without protein, we would lose a lot of bodily function. We also have to think proteins are responsible for cell synthesis and repair. So if something goes wrong in our cells, proteins are the ones to make it go right again. If we need to repair a cell, proteins are the ones to call on. And then from what you probably are most familiar with in terms of protein forms the structural support of our body. Without muscles, which are primarily protein, we would be human skeletons walking around. We wouldn't actually be walking around because we couldn't walk without muscles. So protein is extremely important in that structural support of our system as a whole. If we think beyond the body, protein does have quite a good function in food. It provides structure. And I left it at just as structure, but there are many forms of protein that include emulsification factors, gelatin factors, even in bread dough, we're getting the structure from a protein. So structure is a very general term to represent texture or structural component of foods, especially through the cooking and the heating process. So on a very basic level, proteins consist of atoms, just like carbohydrates. Like carbohydrates, they have carbon, hydrogen, and oxygen. But we're going to go a little bit further and say proteins also contain nitrogen. That is the basic definition of protein. It contains nitrogen. And some proteins contain sulfur. Beyond that, away from those primary atoms, we see some proteins have iron, copper, phosphorus, and zinc. Those are specialized to certain amino acids and certain protein makeup. But if we think about proteins beyond the level of atom, level being the smallest basic level, once we put atoms together, we make things called amino acids. So amino acids are the basic structural unit of a protein. If we put amino acids together to the next level, we form polypeptides. And polypeptides are the more tangible And I say tangible, we won't ever really feel proteins on an individual unit, but the larger subunit that help us create muscle in our body or enzymes that help us function. So let's talk about those amino acids to start with. All amino acids have a basic structure. There are 5 components to that basic structure, and I'm going to draw you through this basic structure just so you're very clear on how we put that structure together. So first we have a central carbon represented by a capital C. Every carbon atom in our universe can bond to 4 different things. So I put 4 lines on here to represent 4 molecular bonds or chemical bonds. The first atom that carbon is going to bond to is hydrogen, represented by an H. Just one single hydrogen atom. Very simple. Next, the central carbon, is going to bind to what we call a carboxyl group. I can say carboxyl group. I can also say a COOH group. I can say a COO minus group, or I can say an acid group. The carboxyl is an acid and allows functionality of amino acids. That's where the amino acids gets its name. Then thirdly, it binds what we call an amine group. Whenever you hear the term amine, you should think nitrogen. So remember that nitrogen allows us to define an amino acid. Without nitrogen, we do not have an amino acid. So we bind a nitrogen group to that central carbon. So we get the term amino acids from the fact that central carbon binds an amine group with the NH3 and a carboxylic acid with the COOH group. And then lastly, the 4th bond is represented or bound to an R group. And I say R group in very general terms. That R group can change based on what amino acid we are talking about. So the R is the identifier for the amino acid. Every amino acid has an amine group, NH3. Every amino acid has a hydrogen group, H. Every amino acid has a carboxyl group, the COOH. But individual amino acids vary based on the structure of their R group. So my R group would be Mackenzie. That's my identifier. Your R group might be Michelle. That's Michelle's identifier. Someone else's R group might be Jake, and that's Jake's identifier. Okay? So if we look at those R groups, you can see they are highlighted in either yellow, green, blue, or purple. Those R groups change from amino acid to amino acid. So if we look at glycine, we see that glycine has a central carbon. I'll point to these. A hydrogen, an amine group, an NH3, a carboxyl group, a COO or a COOH. You'll notice the H has been dropped from this one. That's fine. It's still representative of a carboxyl group. But glycine has this R group that's simply just an H. So glycine is the smallest molecular weighted amino acid because there's only an H. If we move to alanine, we see that there is a CH3 and there's another carbon associated with that central carbon. So these R groups can be very complex. We can have some ring structures like we see in tryptophan or phenylalanine. We can see double bonds and single bonds. We can see additional nitrogen groups. So that R group is the identifier for our amino acids. And that R group also expresses the character of those amino acids. So we have non-polar amino acids, polar amino acids, positively charged amino acids, and negatively charged amino acids. Don't worry about the classification for our class. You will learn about that in future classes, or if you're a senior, you've already taken classes, these should be familiar to you. Just know that the R groups change from amino acid to amino acid. So now we can talk more about protein structure. Remember, proteins are polypeptides of amino acids, meaning proteins are individual amino acids that are linked together to form some sort of shape. And that shape comes in 4 different levels. The first is primary protein structure. Primary protein structure is what starts it all off. Once we have primary structure, that protein can start to develop secondary, tertiary, and quaternary structure. So in class on Wednesday, you will develop a model in front of you that you can touch, feel, look at, whatever you need to determine or differentiate primary, secondary, tertiary, and quaternary structure compared to other groups. Okay, so here I've got a depiction or kind of an analogy. Primary structure is what we consider letters in the alphabet. They act on their own, and if we string them together, we can make words, and that would be secondary structure. Then from words, we can make sentences, representative of tertiary structure, and then from sentences, we make paragraphs, quaternary structure. So I'm going to go through each of these in more detail. Starting with primary structure, it's essentially a string of amino acids put together. So you remember how when we talked about amylose was a single file line of glucose molecules. Here you can think of primary structure as a single file line of amino acids put together. Those amino acids can be one of 20 amino acids that are found in our body or other mammalian bodies. So as a meat eater, we are eating primarily just 20 individual amino acids. Their primary structure differs, though, because they are strung together in different orders. When we combine two amino acids, like we're doing on this slide, we have two glycine molecules. When we combine those together, a chemical reaction occurs that allows for loss of water, you'll see that minus H2O next to the arrow, to form a peptide bond. That peptide bond formation is a condensation reaction because we've lost water. So our body gets some water from the chemical reactions that are happening within us. Now, it's not near enough water to keep us hydrated, so we still have to drink water during the day, but that is a source of water for us to use for bodily function. Once we put two individual amino acids together, put two of them together, we get a dipeptide. If we continue to add amino acids to each end of that dipeptide, we will get a polypeptide, meaning many amino acids linked together. And that is primary structure. Once we have many amino acids linked together, those amino acids, particularly the R chains or the side groups, and the hydrogens associated with them, will hydrogen bond to each other and create two different structures. One called beta pleated sheets, which you'll see down here. You'll see that pleated structure, and alpha helices. So it's like a staircase, a spiral staircase. And you'll see these little 3 dots between an H and an O on individual amino acids. That's the hydrogen bonding that's occurring. So we can get beta pleated sheets stacking on top of one another, or we can get that hydrogen and oxygen from our amino acids starting to hydrogen bond in an alpha helical structure. You will see alpha helices and beta pleated sheets in most all proteins. There's a combination of. It's all dependent on what amino acids are interacting in that structure. Next is tertiary structure. This is where we start to see 3-dimensional folding patterns start to occur, again based on the side chain interactions or those R group interactions. Once we start to see 3D, we know we're at least in tertiary structure. You'll see in this image there are 4 different things that cause proteins to start folding around each other. That would be hydrogen bonds, just like we saw in pleated sheets. Disulfide bonds, we know that proteins contain sulfur. Okay, so if you ever smell rotten egg smell, it's because these disulfide bonds are being broken and releasing an aroma. We also get hydrophobic interactions where proteins are starting to repel each other based on their chemical nature. And we can get salt bridges. So don't worry about these 4 different bonds. Just know that's interaction again between R side chains to cause a 3-dimensional folding pattern in protein structure. Then finally, we can take that 3-dimensional folded pattern and combine them with other 3-dimensionally folded proteins and get quaternary structure. This is when 2 people come together to make a team. This is when 2 proteins come together to make a team. So you'll see in the bottom right-hand corner, we have 4 different colored proteins. They've all started to bond together and create quaternary structure for that protein as a whole. All right, I know that was a lot on protein structure. In class on Wednesday, you should be comfortable with that because we're going to build protein models. Now let's kind of go more into protein as a food, because that's really what we're about. Biochemistry is beyond us. We're just talking food here. When we think about amino acids, remember the body has 20 amino acids that we use to function. There are what we call essential and non-essential amino acids. Essential meaning we must consume them with our food, okay? So we cannot make those essential amino acids. Our body cannot do that by itself. So we have to consume foods that provide us those essential amino acids. The non-essential amino acids means we could sit here and not eat a thing and our body would still make those amino acids. Okay, so there are 9 essential amino acids, and I put an asterisk by that 9. There are some sources that say there are 10. Some say that there are 9. And if you look at the list I have on the right here, I list 10. Okay, so for our class, we're going to say there are 10 essential amino acids. So you can cross this 9 out and put 10. And the reason we're doing that is because we are including arginine in the group. Arginine is only essential during infancy, it's definitely essential during infancy, but we don't necessarily make enough arginine for our body to use fast enough. So our bodies can make it, therefore it is non-essential, but our bodies don't make it fast enough for us to use efficiently. So consuming foods with arginine is important. Same thing with histidine. Histidine, however, is definitely only essential for infants. Once we reach a certain age, our body has developed the function of making histidine, so it becomes non-essential. But as a comprehensive list, remember there are 10 essential amino acids. We are including 10 in this class. Other sources include 9, mainly because arginine is kind of the one that comes up in a question. So why can our body not make these? Why do we have to eat them? Well, there are a couple reasons. First, our cells cannot make the carbon skeleton. So we can't take a single carbon and create those 4 bonds specific to these R group essential amino acids. We just don't have the functionality there. Or our body cannot attach the NH3, the amino group, to the carbon skeleton for these amino acids. Or finally, we cannot synthesize that skeleton fast enough to meet our needs. And this would be the case where arginine comes into play. So foods that are providing all of the essential amino acids to us are considered complete proteins or high quality proteins. So there are oftentimes on labels that you will see a source of high quality proteins or a source of complete proteins. That means all 10 of these essential amino acids are included in that food. Now, if you will be responsible for knowing these or memorizing these 10 essential amino acids, there is likely a chance, especially if you're a science major, you will need to know these in the future. An easy way to do that is through this mnemonic, PVTTIMHALL, otherwise known as Private Tim Hall. So the phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, and lysine. Okay, so some of you may have heard that mnemonic before, some of you may be new. It's just an easier way to remember those 10 essential amino acids. So I mentioned complete versus non-complete protein. A complete protein is a protein that provides all of the essential amino acids. Any animal-derived protein, with the exception of gelatin, is a complete protein. So meat, poultry, fish, eggs, milk. Gelatin, during the processing of gelatin through heating, we lose one of the essential amino acids, and it becomes low in others, so it's not quite a complete protein. Anything that's a non-complete protein is probably plant-derived. Plants are low in at least one of the, remember for our class it's going to be 10 essential amino acids. The exception would be quinoa and soy protein. That is why soy protein is very popular in vegetarian diets, because it's a complete protein. That doesn't mean that certain foods that are incomplete protein sources cannot be consumed. because we have what we call complementary proteins. So that might be two plant-based proteins that are incomplete, can be consumed together to compensate for any amino acid deficiency. So as an example, black beans are low in methionine and tryptophan, so they're incomplete proteins. But if we eat them on a flour tortilla, say in a taco, that tortilla is going to contribute methionine and tryptophan. So as a meal, as an entire meal, we see a complete protein. That flour tortilla is also low in lysine, but the beans contribute lysine. So it's kind of a give and take. I'll be your friend if you be my friend type of thing. We also have to remember that those two incomplete proteins, if eaten together, don't necessarily have to be eaten in the same meal for your body to be sufficiently provided complete proteins. It can be through the day that you maybe eat black beans in the morning with your quinoa healthful breakfast, and then for lunch you eat flour tortilla on a taco. Then there are what we call conditionally essential amino acids. So we have amino acids that become essential to our body under certain conditions. One of those conditions would be the disease phenylketonuria. PKU is what I'm going to refer to it because it's a very big word for me to say. Phenylketonuria, I'm not going to try. PKU occurs, it's a brain and neurological disease. It's when an enzyme that converts phenylalanine to tyrosine doesn't work. So if we're not converting that phenylalanine to tyrosine, our body becomes deficient in tyrosine. So tyrosine becomes a conditionally essential amino acid. We have to consume it. Another example would be during wound recovery. Glutamine stimulates inflammatory response. So we need inflammatory response to heal wounds. So if we have large wounds, say during wartime, glutamine becomes a conditionally essential amino acid. So now let's think on a broader scope of protein. Where do we get protein? The most common answer is from animal products: beef, chicken, poultry, pork, fish, seafood. So animal products are going to provide 70% of North Americans' diet's protein. Plant products also are plenty fine enough in protein. They may not be complete proteins, but there's still amino acids found in them. Plants contribute 65% of the protein worldwide. So we in the United States and in other populated kind of modern countries are fortunate that meat is readily available for us. However, in countries that meat is not a staple food, we're still seeing protein efficiencies. Protein is not a deficient nutrient because plants provide protein as well. Then lastly, I'm going to finish with what we call biological value, or the BV. BV, in the simplest term, is the efficiency of food protein to be absorbed and converted to body tissue. So any food you eat that has protein has amino acids, and those amino acids have a nitrogen or an amine group associated with them. If those amino acids mimic the makeup of your body, then we are saying that's a high biological value food. So high BV foods would be animal proteins, because animal protein sequences or structure more closely matches our body's protein structure as compared to plants. So BV scale is from 0 to 100. Egg white protein is the 100. It's the gold standard for biological value. Almost all of the amino acids in egg whites will be absorbed in our small intestine and utilized for body tissue. So we can grow muscles, we can continue to regenerate skin cells if we eat high biological value foods such as eggs. If we think about it, animal proteins, beef I believe is around the 80-85 mark. So still high, but not quite as high as 100. So some portion of the amino acids found in meat products will be excreted as waste. We don't absorb all of them. Plant proteins, we absorb lesser amounts, and it all depends on the plant that we're eating. But on a general level, plant proteins have a lower biological value than animal proteins, the exception again being soy proteins. So that's why vegetarian and vegan eaters incorporate a lot of soy into their diet, because it's a complete high biological value protein that's going to contribute to bodily function. All right, that's a wrap for today's pre-party. We will see you on Wednesday. Be ready to build protein models.